Okay, so we're going to get started with Module 57, which is in Chapter 17, and this is going to be on toxicology and chemical risks. So again, we're talking about human health here. So this one's going to be, the last one was on viruses, pandemics, things like that. This is going to be on things that can harm people and affect human health in the environment. So after reading this module, we should be able to identify the major types of harmful chemicals, explain how scientists determine the concentrations of chemicals that harm organisms. You can see here a list of major concerned chemicals from lead, which used to be found in paint and gasoline. It's definitely been, that's why your gas is called unleaded now. It's a neurotoxin that impairs learning, nervous system disorders, and can lead to death. Mercury. Mercury is found in coal. We do try to remove it from coal now. And it also bioaccumulates in fish. And so fish consumption can lead to mercury poisoning, which is a neurotoxin, and can damage the brain, kidneys, liver, and immune system. Arsenic is a big one. This is found, this comes from mining operations and can leach into groundwater, and this can cause cancer, as well as asbestos, which used to be used in building materials, and is a carcinogen as well, that can lead to lung cancer and impaired breathing. PCBs, or polychlorinated biphenyls, are an industrial chemical, which can be a carcinogen, leading to liver damage. Radon, which can be found in your soil and water, lung cancer. And you can see more of them down there. Even alcohol, which can lead to reduced fetal growth. Other ones, which are pretty much pesticides and insecticides and stuff that's found in cosmetics. And those ones are endocrine disruptors, which then impact reproduction and fertilization. So chemicals that harm organisms are classified as a few different ways. Neurotoxins are ones that disrupt the nervous system of animals, the brain function. Carcinogens are ones that cause cancer. An mutagen is a type of carcinogen that causes damage to the genetic material of a cell. So that's a specialty type of harmful carcinogen. Teratogen is a chemical that interferes with the normal development of embryos or fetuses, thus affecting, this is like what alcohol could do, can affect the development of a fetus in a body. An allergen is a chemical that causes allergic reactions, and endocrine disruptors are ones that affect with the normal functioning of hormones in the animal's body, and can even lead to the loss of ability to reproduce. So scientists can determine, though, the concentration of chemicals that harm organisms. So to assess the risk of a chemical, we need to know the concentration that causes harm. There are 3 different ways to determine the harmful concentration of a chemical. We look at dose-response studies, which are the ones that you're pretty much going to do in a lab. We're going to look at prospective studies and retrospective studies. A dose-response study is a study that exposes organisms to different amounts of a chemical and then observes a variety of possible responses, including mortality or changes in behavior or reproduction, to see how giving different doses of that chemical actually affects those organisms. We tend to want to do this. This is necessary to see how a chemical is going to affect living organisms, and then kind of scale that up to humans. It's not exactly a great model for humans all the time. We do try to do this as much as we can with invertebrate animals, like insects and things like that. But there will be like rat and mice studies on this as well. Acute study is an experiment that exposes organisms to an environmental hazard for a short duration, just like we talked about acute diseases and chronic diseases. You've got acute studies, which give you a large amount for a short amount of time, and a chronic study, which would give you a little amount of an environmental hazard for a long duration. And so all of these lead to this number, which is called the LD50. and that's going to be the lethal dose of a chemical that kills 50% of the individuals in a dose response study. So let's say you had fruit flies, you're exposing them to lead, and you gave them different concentrations of lead. At some point, there would be half of the fruit flies that perished. That would be, once it hit 50%, that would be the concentration that was the LD50. That was the lethal dose that kills 50% of the population. And that's a big number that scientists like to use in order to describe the harmful effects of different concentrations of chemicals. You can see right here. So scientists will expose animals to different doses of a chemical. So you start, this is just hypothetical units. You can say concentrations, milligrams per liter, or whatever you want to say. And as it goes up, this is your percent mortality. As you get higher and higher, your mortality will go up. Once you hit 50%, right there, this, which would be about, in this case, 5.5, would be your LD50. And then there would be a point where like 99% of the participants died. Typically, dose response studies produce that S-shaped curve. The sublethal effect is the effect of an environmental hazard that's not lethal, but which may impair an organism's behavior, physiology, or reproduction. So not every chemical is going to kill the population, but it can severely harm the animals in there. So instead of an LD50, sometimes we'll look at an ED50, which is the effective dose of a chemical that causes 50% of the individuals in a dose response study to display that harmful effect, whether it be a mutation, whether it be like a heart defect, or any kind of thing, maybe a reproductive effect, sterility, stuff like that. The ED50 is going to be the effective dose. What's the concentration of this chemical that causes 50% of the population to display this harmful trait? So now outside of the lab, what we'll look at is retrospective studies and prospective studies. So what a retrospective study would be, this would be something where you might see the case in Flint, Michigan, where we know people have been exposed to, harmful stuff, the lead that they've been exposed to there. And so the study is going to monitor people that have been exposed to an environmental hazard at some time in the past to see how that affects them in the future. And see how it's affecting them right now. So that'd be retrospective because they were exposed in the past. Now, prospective study is going to be a little bit different. It's going to be looking at people who may be living in an area that we don't know if it's harmful or not. Maybe it'd be like some different areas around Chernobyl, or places that have had some kind of environmental disaster, or maybe there's mining nearby. Scientists will study those people to see if being in that area or being exposed to things in that area is affecting them. And so that's a prospective study, looking at, they haven't been exposed yet. They will know that there are inherent risks there, and that scientists are going to be looking at them. They don't put them there or anything like that. They just want to see, is living in this area harmful? And a synergistic interaction is a situation in which two risks together cause more harm than expected based on the separate effects of each risk alone. So sometimes chemicals can actually combine together and produce a worse effect than if you just had those two chemicals separate and they became together. They can work together basically to produce more harm. So factors that determine the concentration of chemicals that organisms experience. So to identify and understand the effects of chemical concentration, we need to know something about how the chemical behaves in the environment. So this is where we'll look at the route of exposure, which is the way in which an individual might come into contact with an environmental hazard. So if we know lead is harmful, how are people going to get lead in their system? Is it coming from the pipes that the water is being transported in? Is it coming from the paint they're using in their houses? Is it coming from the lead, from the gasoline that they're pumping into their cars? What is the route of exposure? So there's a multitude of potential routes of exposure in most chemicals, but despite that, most chemicals have a limited number of major routes and then we can trace it back. So some chemicals can get in through your food, some can get in through the air, some through the water, some through the soil, some from mother to fetus, that type of thing. So the solubility of chemicals, bioaccumulation, and biomagnifications. The movement of a chemical in the environment depends in part on its solubility. Hopefully you remember that from chemistry. Solubility is how well a chemical or a solid chemical or even a liquid chemical dissolves in water. A water-soluble chemical can be washed off surfaces, percolate then into the groundwater, run off the surface into waters including rivers and lakes, and then if it's very soluble in water, if it dissolves in water, that will then be ingested by humans who use that water. So fat-soluble chemicals are not very soluble in water, are found in higher concentrations bound to soils, including benthic soils that underlie bodies of water. So these can even be in soils, not just the water. We need to talk about bioaccumulation, biomagnification. Bioaccumulation is an increased concentration of a chemical within an organism over time. Biomagnification is bioaccumulation is, let's say that there's a certain amount of a toxin in grass and cows go and eat that grass. Well, over time, they're going to accumulate more and more and more of that toxin. Then biomagnification is, so you have a certain amount of concentration now in that cow. Now, if a human eats a bunch of cows that have eaten that grass, they're going to, bioaccumulation now is going to be magnified like tenfold of what was in that cow. So that's called biomagnification. It's the increase in chemical concentration in animal tissues as the chemical moves up the food chain. And persistence is important because it's the length of time that a chemical remains in the environment, and that can tell you if what you're eating or what you're consuming is, if it's been exposed to a chemical, is safe or not. So you can see right here the biomagnification of DDT. DDT was a pesticide used. It ran off into water, very low concentrations. The plankton, though, continually consumed water that had a low concentration of DDT. And so their concentration was way higher because they were continually consuming it and was getting into their system. And so because they were continually consuming it, their concentration is way higher than the concentration that was in the water. So then, same thing with the fish. So smaller fish would eat the plankton and continually consume a low concentration of DDT in the plankton, but would continually get it, so they had magnified. And then it magnifies into flounder, which is a larger fish, and eventually it'd get up to the osprey. So when the osprey, though, eats 1.28 parts per million in each flounder, all of that's going to accumulate. It's not going to come out of their system, so it's going to accumulate and accumulate and accumulate. So the higher you up on the food chain, if something is being contaminated, the more magnified it's going to get. And this has actually caused, different populations of birds and predators like that to actually get pesticide poisoning because it had moved up and magnified up the food chain. So let's look at some persistence. You can see different half-lives. A half-life is the amount of time it takes for a chemical to be reduced in half. So you want something that's a pretty quick half-life in order for it to degrade. So this insecticide, malathion, is a very short half-life. You put it on, half of it's gone, half of it's degraded in a day. But it goes all the way up to DDT that we just talked about, super harmful pesticide that was used, an insecticide, can stay up to 30 years in soil, so that's pretty bad. You can see other ones here, atrazine, Roundup, those are both herbicides, and look at that, 224 days. And wetland soils, PCBs, used in industry all the time, 8 to 15 years in water, so pretty persistent chemicals there. And that's it for Module 57. We'll be in Risk Analysis Module 58 in the next lecture.